I will tell you today something about the introduction, or I will introduce about the foundations of stem cell reprogramming and will try to give you insights into the newest and the most efficient methods for this. So let me briefly go into the agenda for today. First, I like to start with an explanation what stem cells actually are. Then I want to introduce the technique of reprogramming and tell you what it is used for. I will introduce different reprogramming techniques and will go further into detail for one technique, namely the mRNA reprogramming. And the last point for today will be the verification of iPSCs. So let's start with the first topic. What are stem cells? So when we talk about a stem cell, what do we mean by that term? Stem cells are defined by two characteristics. The first is their ability to self-renew. That means that they are able to create an identical copy, and this way they can be maintained in a culture. So they build a constant pool of stem cells for regeneration. The second ability is the capacity to differentiate into specialized cell types. Usually progenitor stages that are more restricted in their developmental potential and will then differentiate into further specific lineages. So here I want to describe shortly the development of an embryo and show where to obtain embryonal stem cells. So the whole story begins with an oocyte which is fertilized by a sperm. The next step in the development will be the formation of a morula. And then the next step will be the formation on about day 4.5 of the blastocyst. And this consists of an outer cell layer, here symboled by these yellow cells, and also a blue symboled inner cell mass. And from this inner cell mass, you can separate embryonal stem cells. So these blue cells are the embryonal stem cells. And these embryonal stem cells has a special feature that they can differentiate into all the lineages of a body. For example, they can differentiate into cells of a circulatory system or the nervous system or the immune system. And if you follow this whole process of development, then this is what is described as differentiation. I also want to introduce other terms to you which you will face if you are going more into detail for stem cell research. There are different features of different developmental stages, and the first feature is the potency, which describes the ability to form all tissues in the body and the placenta. And this is a feature the morula has, for example. And this means that all cells inside the morula are able to not just form the embryo itself, but also extra embryonal tissue. If you go further in the development, the next feature will be the pluripotancy. And this is the ability of the embryonal stem cells, the ability to form 3 germ layers, like here displayed. The next feature is the multipotency, and this is the term for the ability to differentiate into multiple cells or tissue types, the so-called progenitor cells. And an example for this are the hematopoietic stem cells, which can form different cell types of blood, but nothing else. And the last feature in this development is the unipotency, which describes the limited differentiation potential into just one cell lineage, the so-called precursor cells. So as you have now learned where to find stem cells in vivo, how can we produce pluripotent stem cell or pluripotent cells artificially? Here you see again what I told you before. Here we have the pluripotent stem cells, so embryonal stem cells abbreviated by ESC, and they can form these three germ layers of the body. The ectoderm, which consists cells of like skin and brain, the mesoderm, which can form bone and blood, for example, and the endoderm, which forms pancreatic cells and lung cells. And this process from the ES cells to the differentiated cells is called differentiation. And for a long time, it was thought that this is a one-way road, where you could just move forward into a more differentiated stage. But however, especially with direct reprogramming done in the Yamanaka lab, I will explain this to you later on in more detail, and then it became clear that you can go back from a differentiated cell into a pluripotent stem cell stage. And this is forming then, or this process forms then the so-called induced pluripotent stem cells, here abbreviated by iPSCs. Here I want to introduce a timeline to you about the most important publications about reprogramming, and I just want to highlight two of them. The first is that here the first evidence that reprogramming of somatic cells is possible was done in 1962 by John Gurdon, and he cloned frogs by transferring the nuclei of differentiated intestinal cells into amphibian oocytes. And then, pretty much later, in 2006, the Yamanaka lab found out how to mimic this process with transcription factors and maintain these cells in a pluripotent stage. So iPSCs behave like ES cells, for example in self-renewal and pluripotency, but also show a similar morphology, also proliferation, gene expression and epigenetics. So why are iPS cells now so attractive? Because they become, for example, the ethical challenges of an ES cell, as you always have to destroy the embryo if you isolate the inner cell mass of the blastocyst. And these iPS cells are especially interesting to the field of regenerative medicine, as it's possible to reprogram patient-specific cells, for example, of a skin biopsy, and they can get then further differentiated to study genetic diseases and screen for therapeutic compounds to find drugs. Because I promise you, it's pretty much better to kill the cells with non-working drugs than killing the patient. The other way around, you can also use, if you have a known disease mutation, you can take this patient-specific iPS cells to repair this iPS cells, then further go on with differentiation, then you have your healthy cells, and this you can use for transplantation into the patient, and this eliminates the danger of immune rejection. So this slide uses the so-called Wonnington's landscape to show the difference between cell states as a sort of an activation energy concept. So once the cell moves down the slope, it differentiates and cannot go back in their normal development. So here you see the second differentiating cells, and it goes a different way. And however, as shown with the second ball, Reprogramming bypasses this and sends a cell to pluripotancy. And note, the ball is not shown to travel back along the same path it took to differentiate. So you would never exactly know where your cell is in this landscape. How this reprogramming process is working in detail is not completely unraveled today, but here I want to make it a little bit more clear. At the top of the picture, the whole story begins with the application of the 4 transcription factors OCT4, SOX2, KLF4 and SemMACS. And this destabilizes the somatic cells, in this case here fibroblasts, and then a big case is occurring. A lot of cells begin to do things which we don't want them to do. Other cells begin to establish certain expression profiles and just a few number of cells. And in this, this is symboled by this bottleneck over here. Just a few number of cells starts to establish the expression profile we wanted to have. A whole cascade of protein expression starts while the epigenetic landscape of the cells change, and if this occurs and the transcription factors shown here are expressed, the reprogramming is done and the somatic cells transformed into an iPS cell. So let's go for the third point. Which reprogramming techniques do we have? So I just briefly introduced to you the first technique, which was the first evidence that somatic cells can be reprogrammed, which has been published in 1962 by the Gurdon lab, and he performed nuclear transfer from differentiated intestinal cells into amphibian oocytes, which leads to cloning of frogs. And they showed that all genes needed to make an organism are present in a specialized cell. So this was, maybe you know the story, this was also done with a cheap dolly, so just the cloning of an animal. The next method is the cell fusion, which is an alternative approach to reprogram the somatic genome, which involves the creation of hybrids between somatic cells and other cells that contain reprogramming activities. And the special feature is, or the advantage is, that the pluripotent phenotype seems to dominate also in somatic cells. So if they divide over here, then always the pluripotent phenotype is obvious. But unfortunately, it has been shown that this method is very inefficient. And the last method I want to introduce is the direct reprogramming, where we have the introduction of a small number of transcription factors, of these Yamanaka factors, and then the cells are reprogrammed. And this is now the gold standard to produce iPSCs, and we will go further into detail during this presentation. So there are 2 different groups of methods which exist for direct reprogramming. One that includes carrier for the transcription factors that integrates into the genome. So here you see that. So later on the genome is altered. And one method that is integration free, and therefore the genomic integrity is maintained. And if we want to go for clinical trials and therapy, we of course should go for the integration free method to conserve the original DNA. Nevertheless, I like to shortly introduce 2 important integrative methods. First is the retrovirus, which has been used by Shinya Yamanaka. But this method takes up to 6 weeks to form iPS cells. Partial reprogramming, so we have two phases of reprogramming. Partial reprogramming generates class 1 iPS cells that express the rival transgenes and endogenous pluripotancy genes, and full reprogramming in so-called class 2 iPS cells silences the vector as the endogenous genes maintain the pluripotent stage. Thus, retroviral vector silencing serves as a beacon marking the fully reprogrammed pluripotent stage. The disadvantage is that you alter the genome and don't have any control about the dose. And furthermore, you don't know if the silenced genes are reactivated in a later stage. Also, the efficiency is very low. It's below 0.1%. And here we have the second method I want to introduce, the piggyback transposon system, which is a mobile genetic element that transposes between vectors and chromosomes via a kind of a cut and paste mechanism. And during transposition, the piggyback transposase recognizes transposon-specific sequences located on both ends of the transposon vector and moves the contents from the original sites and integrates them into specific chromosomal sites. But the special feature of the piggyback transposon, different from the retrovirus, is that it's also reversible. Nevertheless, it is a genetic modification, and again, we have no final control about the time and dose of the expression of these transcription factors. For the integration-free delivery, there are also different methods on the market, and here I want to introduce two of them. The first is, where it is, here. The Sendai virus, which acts exclusively in the cytoplasm of cells and cannot enter the nucleus or alter host chromosomes, and it is therefore fundamentally free of the risk associated with conventional viral vectors. Sendai viral vectors also reprogram cells with high efficiency. The disadvantage of using Sendai virus is that they have relatively high costs compared to other methods. The establishment of virus-free iPS cell lines requires up to 10 to 20 passages, depending on the cell line, and reprogramming conditions, and you must isolate and clonally propagate at least 10 primary colonies to establish only a few iPS cell lines per patient sample. Another integration-free reprogramming method is the mRNA delivery. And here it's very important that you have the ability to reprogram stem cells using a method that is both non-integrative and non-viral. And this is very important for future therapeutic application. And the advantage of this technique is that there is no risk for insertional mutagenesis that can later cause cancer. And for cells that will eventually be used in the clinics, this will likely be the dominant technology. Also, a big advantage is that it's very fast, this technology, and you have a very, very high efficiency, about over 1%. For example, mRNA reprogramming can result in iPS cell colonies within 2 weeks. But one disadvantage of the mRNA reprogramming is the inconvenience posed by the daily mRNA transfections required for this method. All right, so let's go further into detail for this mRNA technique. Warren et al. Published in 2010 how to generate synthetic highly stable mRNA, which can be used to reprogram iPSCs. And in their publication, they showed reprogrammed cell colonies within up to 17 days. So this is what you see here. I also want you to introduce our reprogramming kit, the SemMACS mRNA reprogramming kit, and we will see one typical workflow for mRNA reprogramming. So we start the whole culture with the pre-culture of somatic cells, like for example fibroblasts, in ReproBrew XF medium, and then coat plates with CTS and plate the cells on top, allow the cells to adjust, and on day 0 start with the transfections. We will go on with the transfections for 12 consecutive days, also adding this B18R, which was identified to enable increased cell viability during RNA transfection. And on day 11, after the 12th transfection, we stop with the transfections and allow the iPSC colonies to grow. Then we come to the point where we change the medium to iPS brew medium, and iPS cell colonies are visible. And I want to do that with you, more or less live. And this is what you would see through the fluorescence microscope on day 1. These are fibroblasts, and they have been transfected for 1 or 2 times on day 1. And the big advantage in our mRNA reprogramming kit is that we use EGFP as a transfection control. So if these cells are green fluorescent, like you see over here, this shows directly how efficient this method is. And you see more or less 100% of the cells are transfected. So here you see day 3, the morphology didn't change. They grew a little bit, the cells, but still they are more or less 100% green, so efficiency is still very high. This you can also observe on day 5. Cells grew nicely. And here on day 7 you see the first sign that the morphology is changing a little bit. So here you have more dense parts in your culture. And on day 9 it gets a little bit more obvious. Here there are very dense parts, and you see maybe there's a little bit enhancement of the size of the nuclei, which is normal for ES cells, but still the efficiency is very high. And then very obvious on day 11, this is what you want to get out at the end of reprogramming. This is a nicely formed iPSC colony. To be sure for the pluripotancy of these colonies, you can also stain them with OCT4, which is a transcriptional marker for pluripotent stem cells and serves like a fingerprint to identify iPS cells. And in this case, you see very nicely formed green fluorescent colonies. To keep the iPS cells further in culture, you have to isolate them from the differentiated cells, because you always have, as I showed you before, a lot of differentiate or other cell types in your dish, and also iPS cells, also dead cells. And you just want to go on with your pluripotent cells. And one method would be that you manually pick these cells and isolate them. So it's very hard, I can tell you, to pick that pluripotent cells, as you don't really know if you have really pluripotent cells or not, as you just have your dish and they are not fluorescent or something like that. Because normally for a picture like this, you have to fix your cells and then you kill your cells. So you cannot go on with further passaging. Therefore, you would pick all cells which have a pluripotent morphology. You would replate them and passage them further on. But a new technique or a new highlight in our product portfolio is a live cell stain antibody, which you can see over here. And for this, you don't have to kill your cells and you don't have to fix your cells. And you can directly, under the fluorescent microscope, see what you really are or want to pick. One additional problem is that you maybe assume one colony is monoclonal, so just originating from one cell. But as you see by this picture, it's not every time the case. The more efficient option to isolate the reprogrammed cells is to separate them. And this is one very famous technology for Miltenyi Biotec. Therefore, we have this TRA-160 microbit kit for the isolation of mRNA iPSCs. And this is working like I will explain now. So you have your pluripotent cells, which are expressing TRA-160. And we have very, very small microbeads, which binds to the TRA-160 positive cells. These microbeads are consisting of iron, and if you put that sample with your pluripotent cells and some other cells inside on top of such a column, where is a steel ball matrix inside, and you place the column into a magnet, then these microbeads attached to the cells will bind to the column. And everything what is not positive for TRA-160 will run through. And if you later on release the column from the magnet, then the column is not magnetic anymore and will not bind the magnetic beads anymore. And then you can also wash out your then positive sample and hopefully have just your TRA-160 positive cells. And this we have done over here and also analyzed with our flow cytometer. What is displayed in these plots is two markers. At first TRA-160 for pluripotency, and here the EBCAM marker, which also serves as a pluripotency marker. And here on this part of the blood, you see cells which are negative for both markers. Here you see cells which are positive for both markers, and here you see cells which are just positive for the EBCAM marker, and here you see cells which are just positive for the TRA-160. And what we want to obtain later on are these cells. So in the original fraction, just after the reprogramming, you have, for example, here just 42% of pluripotent stem cells. And if you now place all these cells, also with the negative cells, on the column, you get out a negative fraction first of all, so what is washed out and don't bind to the cell. And here we analyzed how much pluripotent cells are still here in this fraction, and we saw there's just very small loss of cells of 2%. So, and if we then analyze the positive fraction, we see that the cells are pretty much enriched about a value of 94%. So these enriched cells you can then later on use to replate them, and then you can start with a pretty much higher cell number, which will fasten the experiments dramatically. All right, then we come to the last point of my talk today, the verification of iPSCs. So there are different functional assays of pluripotency. First is the embryonic body assay, and these embryonic bodies are generated by ESCs on non-adherent plates, and this is very important. in the absence of self-renewal cytokines. So they differentiate, and after several days of culture, this is resulting in embryonic bodies, which can be individually transferred and cultured further under conditions appropriate to coax development of various cell types, and then stained for a different cell type marker to obtain if they are really able to form all the cells of the body, and which is the evidence that these cells are still pluripotent. The second assay is the teratoma assay, and teratomas are generated by injection of cells into non-obese, severe combined immunodeficient mice, usually intramuscularly or under testicular or kidney capsules. And after 10 till 12 weeks, or also a little bit longer, you have teratomas, and they can be recovered and analyzed using histological approaches to examine as well if you have all the different tissues inside. All right, that's it for the foundations of reprogramming. Let me briefly introduce where you can receive more support on getting an expert in your stem cell experiments. So we offer a 4-day stem cell course in our headquarters in Bergisch Gladbach in Germany, and also in San Diego, USA, where you can learn theoretically and practically to perform a whole stem cell workflow, including reprogramming, cultivation, and differentiation. And maybe you also want to attend one of our next webinars, and then we invite you very much to come to our website, miltenyibiotec.com/webinar, or you can also find more trainings on our website under miltenyibiotec.com/training. So finally, thank you very much for your attention. Please let me know whether there are any questions. Thank you.